Hi, Ellen. Can you help me to share the welcoming slide? Thank you. Okay, we will start our webinar now. Good afternoon. Good afternoon, ladies and gentlemen. Thank you for joining us for this cancer awareness webinar entitled Cancer Care Through COVID-19 and Beyond. Moving on. Next. The School of Medicine has lined up a series of webinar. Sorry, I think there's a technical issue over here. Is everything all right? Okay. Next, for the welcoming part. I'm sorry for the technical issues. We'll be starting our webinar very soon. Okay, can everyone see this welcoming slide? Is it visible?
Yes, Dolly.
Okay. Okay, moving on. The School of Medicine has lined up a series of webinars on health-related topics. And this is our very first on cancer awareness to commemorate the Cancer Awareness Day, which was on the 4th of February. My name is Dolisha, a Year 3 medical student, and on behalf of Taylor's University, I'd like to extend a very warm welcome to all of you. We appreciate you taking time off your busy schedule to join us today. We hope that you will find this program to be fruitful and engaging. A reminder for all participants, if you have any questions during the presentation, you can scan the code now and type in your questions later. We will bring them up during the forum session. The link will be shared in the Zoom chat box. Please use the Slido link to send all questions and not send them in Zoom chat, as we would like to maintain the privacy of the participants. Thank you. Before we start, I'd like to show you a short video from the Union of International Cancer Control.
Dolly, there is no sound.
My apologies. Let me fix that right now.
I'll take over. I can share now. I'll proceed.
Yeah, sure. Thanks, Ellen. watching the video, I believe all of us are able to appreciate the effort of our healthcare workers for cancer care during this pandemic. For today's webinar, we have 5 great speakers lined up for you. And don't forget to stay till the very end for group photo session and special announcements. To officially start our Cancer Awareness Day webinar, I'd like to welcome the Executive Dean of the Faculty of Health and Medical Sciences from Taylor's University, Emeritus Professor Dr. P.T. Thomas, to give his opening remarks. Thank you for taking the time to join us on this meaningful webinar. Welcome, Prof.
Thank you, Dolisha. Good afternoon, ladies and gentlemen, distinguished speakers, students. I never fail to be impressed at events where our students are the MCs and who work behind the scene. Thank you, Dolisha. Ladies and gentlemen, our world has been focused on the COVID-19 pandemic since March 2020 or a little before that. However, the seriousness of cancer has probably been eclipsed by the attention on COVID-19. Cancer is still there. Cancer kills more people. In 2018, cancer killed 26,965 people, whereas COVID-19 in our country from March, or from when we had the first mortality till yesterday, was 1,249. But our attention and the attention of the government and the media has largely been on COVID-19. But that is common. When there is a present danger, attention shifts to that. But I'm glad that this webinar is being organized today because cancer is still a big problem and maybe even can be said to be a bigger problem. In Malaysia, the 3 most common incidents of cancer are in breast cancer, colorectal, and lung cancer, while the mortality is the highest for lung, and then colorectal and breast are closer together. So it is appropriate that the title for this webinar is Cancer Care Through COVID-19 and Beyond, because it is through the COVID-19 that patients' treatments have been affected, that in the video, like we saw, medications had to be delivered because patients are still undergoing treatment. One of the advantages, one of the benefits, or maybe you can even say side effects of the COVID-19 pandemics is the ability to have webinars, and webinars that can get the experts from around the world. And those experts need not be flown in. There's no time for travel. They can speak at 2 or 3 events in a day. And so we are very fortunate that the School of Medicine has been able to gather a group of speakers who are world-class. We have Professor Dr. Yu Ming Wang from the Kaohsiung Changgong Memorial Hospital from Taiwan. We have our Malaysian champion for cancer prevention and patients, Datuk Dr. Saundari Somasundaram, the president of the National Cancer Society, Professor Dr. Ho Guo Fuang from University of Malaya, and our own Taylor's experts, Professor Chong Pei Pei and Dr. Louis Chung Ying. Cancer is a multifaceted disease. It affects not just the patient, but it affects others around the patient. And so our response to the disease should also be similarly multi-pronged. I wish to congratulate Prof. Roslin Odin, head of the School of Medicine, and his academic staff and the students for organizing this webinar, a very timely webinar, and I'm sure we are in for a treat from all the speakers. Thank you very much and wish you all a wonderful webinar. You're muted, Dolly.
Sorry for the issues. Thank you so much for the opening remarks, Prof. Thomas. So, without further ado, we will introduce our first speaker, Prof. Dr. Chong Pei Pei. She graduated from the University of Manchester with a first-class honours degree in biochemistry and later went on to pursue her PhD at the same university. Prior to joining Taylor's University, she served with UPM for many years and held the position of Acting Deputy Dean of Research at the Faculty of Medicine and Health Sciences. She has been actively involved in collaborative research such as links between genetic polymorphism and cancer risk for virus-associated cervical cancer, and more recently, prostate cancer. She also worked with a National University of Singapore team to unravel the role that the HPV virus plays in modulating a class of enzymes known as ApoB, which leads to mutation and later on to cancer initiation, and is postulated to be a crucial step in the development of subsets of breast cancer. Today, she'll be sharing on the topic microbes and cancer, friends or foes. Now, I invite Prof. Chong for her presentation.
Sorry, Dolly, I cannot hear the volume.
Sorry for technical issues. I just realized that there's no voice. I'll be sharing it again. I apologize for the inconvenience.
Good afternoon, distinguished guests, colleagues, and all participants. Firstly, I would like to thank the organizing committee for giving me this opportunity to speak at this webinar. The title of my talk today is Cancer, Microbes and Cancer, Friends or Foes. These are the contents outlined for my talk, starting from cancer and its multiple causes, zooming into microbes associated with cancers and how do microbes cause cancer. Then I will focus on the human papilloma virus, how it causes cervical cancer and its role in breast cancer development via the induction of the ApoB3B overexpression. Next, I'll move on to the microbiota and microbiome and explain how commensalism by bacterial colonization comes about. Moving on to the microbiome-mediated immune modulation, gut microbiota and cancer immunotherapy, and then end my talk with implications for clinical applications. The central question of my talk is whether microbes are our friends who could help us become stronger and fitter, such as the probiotics which are used in food and as supplements, or are microbes our foes or enemies whose presence in our body causes us to become sick and unwell. Now, cancer is a disease that has plagued humans for decades, and we are still constantly trying to understand its causes and discover new ways to fight cancer. It is now universally believed that cancer is a disorder with multifactorial causes. These include exposure to environmental pollutants and free radicals over a prolonged period of time, imbalanced diet and nutrition, which, for instance, cancer cells are found to feed on high levels of sugar. For lifestyles, a sedentary lifestyle and habits like smoking can predispose a person to higher risk of getting cancer. Genetics play a big role as well in cancer predisposition, whereby some single nucleotide polymorphisms or SNPs in tumor suppressor genes, DNA repair genes, and cell cycle regulation are associated with higher risk of cancer. Hormones, stress, and aging are also factors that contribute to cancer development. And last but not least, microbes, certain microbes have been proven to have a strong causal relationship to cancer. So let's look at some of the more famous microbial culprits of cancer. Number one is Helicobacter pylori, a spiral-shaped bacterium which has been demonstrated to cause peptic ulcer and also gastric cancer. The physician and scientist team of Barry Marshall and Robin Warren were awarded the Nobel Prize in 2005 for discovering the link between this bacterium to peptic ulcer and also cancer. Number two is the human papilloma virus, which has been found to be present in more than 99% of cervical cancer tissues, and also in some cases of anal genital cancers and head and neck cancers. It was discovered by Harold Zohausen, and he was later awarded the Nobel Prize in 2008. The third virus is hepatitis B and C viruses, which have been demonstrated to be linked with development of liver cancer. Besides the three which were described in the previous slide, there are other microbes which were found to be highly associated with different cancers. The oncogenic viruses, which are viruses that can lead to development of cancer, include HPV, which I explained just now, Epstein-Barr virus, the hepatitis B and C viruses, human T-cell lymphoma virus 1, Merkel cell polyomavirus, and Kaposi's sarcoma virus. Oncogenic bacteria includes the main species Helicobacter pylori, while oncogenic parasites include S. hematobium, O. viverrini. Good afternoon, distinguished guests, colleagues, and all participants are also factors. So let's look at demonstrating to the extent of and microbes which were found to be highly associated with different cancers. The oncogenic viruses, which are viruses that can lead to development of cancer, include HPV, which I explained just now, Epstein-Barr virus, the hepatitis B and C viruses, human T-cell lymphoma virus 1, Merkel cell polyomavirus, and Kaposi's sarcoma virus. Oncogenic bacteria includes the main species Helicobacter pylori, while oncogenic parasites include S. hematobium, O. viverrini, and C. sinensis. The link between fungus and cancer development is not so well demonstrated. However, Aspergillus species have been shown to have some contribution towards liver cancer through a toxin that it produces. And recent studies showed Malassezia species was able to cause a certain type of pancreatic cancer. What then is the mechanism whereby microbes can cause or lead to cancer? So we have two possible mechanisms, direct and indirect. The direct carcinogenic pathogens cause cancer via the expression of viral oncogenes that disrupt cell cycle checkpoints, inhibit apoptosis, and contribute to cell immortalization. Examples are HPV, the HDLV1 Epstein-Barr virus, Merkel cell polyomavirus, Kaposi sarcoma virus. On the other hand, indirect carcinogenic pathogens do not induce expression of oncogenes, but instead their persistent infection over a long period of time leads to a chronic inflammatory state or immunosuppression that limits the anti-tumor immune surveillance mechanisms. Examples are HBV, HCV, H. pylori, and the parasites. Another mechanism of indirect carcinogenesis is the production of toxins by the microbes, which could lead to cancer. For example, aflatoxin by the fungus Aspergillus was thought to cause the liver cancer. CAG-A toxin produced by the bacterium H. pylori, and also toxin produced by gut bacterium Bacteroides fragilis was implicated in bowel cancer recently. So how does HPV cause cervical cancer? It has to do with persistence of infection, which leads to accumulation of DNA mutations. So in the initial stage of HPV infection, the virus infects the basal cell of the epithelium, and it can linger in a dormant stage. But after weeks, the virus could replicate and starts to infect the upper layer. But fortunately, at this stage, our immune system is still able to eliminate the virus, and indeed, about 90% of women heal within 2 years. However, if the infection persists for 10 to up to 30 years, then there is a chance for the HPV DNA to get integrated into the host cell DNA. And if it gets integrated into key genes and mutates the genes, it could lead to cancer. So in fact, about 0.8% of the women develop cancer, and some develop invasive cancer. Harold Zellhausen managed to demonstrate that HPV DNA was found in large majority of the cervical cancer cells. Now, it is a well-known fact that cancers are characterized by deleterious mutations in various key genes involved in cell cycle control, but the molecular mechanisms that lead to these mutations were still unknown. In 2013, Burns and colleagues found that a class of enzymes known as ApoBac3B, which codes for the DNA cytosine deaminase enzyme, accounts for up to half of the mutational load in not only breast cancers, but also bladder, cervix, lung, head and neck cancers. They found that the ApoBac3B is highly expressed or upregulated in these cancers. And their findings were published in the prestigious Nature Genetics journal. ApoBac3B is a big family which has 11 members. It's a molecular driver of mutagenesis for cancers. And these enzymes catalyze the deamination reaction, removal of the ammonia group from the base cytosine C to form uracil in the single-stranded DNA substrate. So based on the landmark publication by Burns and colleagues, Professor Naoki Yamamoto, whom I worked with when I was attached in NUS Singapore, he wanted to find out if the HPV virus had any role to play in this. So the hypothesis that viruses are associated with breast cancer was still debatable at that point. But Prof. Yamamoto was convinced that they are somehow responsible because in mice, the mouse mammary tumor virus was found to cause breast tumors. However, molecular epidemiology studies in humans showed conflicting results for the presence of HPV in breast cancer cases across the world. So Prof. Yamamoto hypothesized that HPV could trigger the ApoB3B upregulation and lead to genomic instability, which eventually leads to breast cancer. So we looked at over 200 breast cancer tissues in Singapore, and lo and behold, high-risk HPV DNA was detected in 31% of all the breast cancer samples. And the breast cancer tissues, which are positive for estrogen receptor, showed a significantly higher HPV prevalence than the estrogen receptor negative tumors. Hence, we did some in vitro as well as in vivo experiments and established that HPV oncogenes E6 and E7 could activate ApoB3B expression. Next, we used two techniques, namely the immunofluorescence staining and also Western blot, to show that the cells that are stably transfected with HPV-18 had genomic instability, as shown by the gamma H2AX detection, which is a sensitive biomarker for DNA double-strand breaks. We also performed some other experiments, including the knockdown of E6 and E7 oncogenes and ApoB3B genes in the cells, which were stably transfected with HPV-18. And we demonstrated that DNA damage could be induced through two possible pathways, which are HPV-18, E6, E7, together with the A3B, ApoB3B, and secondly, the virus alone without the independent of the A3B. In summary, our results suggest that HPV and estrogen receptor play a role in initiation stage of breast cancer through activation of ApoB3B. Now, up until 1980s, scientists have been working on single microbes at a time. In 1988, Whipps and colleagues coined the first definition of the term microbiome, which is a combination of the words micro and biome, and meaning a characteristic microbial community. Microbiota, on the other hand, is a term that includes all the microbes like bacteria, viruses, yeast, fungi, protozoa, and archaea that inhabit a biological system. So they are collectively known as microbiota. In contrast, microbiome nowadays refers to the collection of genes within the commensal microbiota. Commensal means to coexist in a mutualistic relationship whereby one species obtains food or other benefits from the other species while benefiting the fitness of the second species without harming each other. So in terms of the human microbiome composition, bacteria are the most abundant and include members of the phyla Firmicutes, Bacteroidetes, Proteobacteria, Actinobacteria, and Myricomicrobia. Fungi are significantly outnumbered by bacteria, and they are many members of the phylum Ascomycota. Viruses, largely composed of bacteriophages, along with some eukaryotic host viruses. For the helminths or parasites, nowadays, we are mostly free of helminths, especially in the developed nations. But some parasites are still colonizing billions worldwide, especially in the less developed nations. including the flatworm, tapeworms, and roundworms. So alterations in the normal microbiota has been reported to contribute to development of many diseases, and some specific bacteria have been demonstrated to be involved in the process of carcinogenesis. Microbiota commensal, especially bacteria, commensal colonization. We know that healthy human body is comprised of approximately 30 trillion cells and is inhabited by approximately 39 trillion bacterial cells. The initial microbiota exposure may occur as early in utero, in the womb, where the gastrointestinal tract of the fetus may first be colonized by maternal bacteria, the mother's bacteria, through ingestion of amniotic fluid. The microbiota becomes shaped by the selective pressure of site-specific factors as the child is growing. And by 3 years of age, an adult-like intestinal microbiota dominated by Firmicutes or Bacteroidetes is then established. The relative proportions of microbiota may vary between individuals and between different anatomical sites like gut, skin, mouth and throat, stomach, vagina have very different composition of the microbiota. The metabolites produced by microbiota play diverse roles in humans, and fermentation and production of vitamins K and B and short-chain fatty acids by the intestinal microbiota occur in the colon. So it's been known that a common set of metabolic pathways is the major characteristic of healthy microbiota among the healthy individuals. The gut microbiota has been shown to have impact on not only local but also systemic and immune-specific programming, immune programming. So experiments involving germ-free mice or GF mice show that they have deficiencies in the GI immunity and they are more susceptible to infections, and these deficiencies can be corrected by colonization with commensal bacteria. Also in GF mice, the mucus layer, which is the first line of defense against pathogens in the intestine, is thinner and has a different mucin composition. They also have decreased number of LP-CD4 plus T cells, plasma cells, and decreased IgA production, leading to further impaired intestinal barrier function. In terms of the systemic immunity, the systemic effects are attributed to circulating bacteria-derived molecules, such as the microbe or pathogen-associated molecular patterns. Lipopolysaccharide, peptidoglycan, or flagellins produced or found on bacteria can be recognized by the host pathogen recognition receptors or PRRs, and this will enhance the systemic innate immunity. Short-chain fatty acids, which are the products of dietary fiber fermentation by the microbiota, have been implicated in stimulating dendritic cell generation. In terms of immune programming, the GF mice has a bias towards the type 2 T helper cell allergic responses, which can be corrected by colonization with commensal bacteria. And these two bacteria, Bacteroides fragilis and Clostridium clusters, can strongly induce the Treg differentiation. And a higher Bacteroidetes to Firmicutes ratio resulting from a high fiber diet can increase the levels of circulating short-chain fatty acids and alleviate the Th2 cell-mediated allergic airway inflammation. Since we now know that microbes can affect the immune system, the next question will be, is there any relationship between the gut microbiota and cancer immunotherapy? An important achievement in cancer treatment in the last 10 years has been the introduction of immune checkpoint blockade, or ICB, immunotherapy, including the use of the anti-CTLA-4 and anti-PD-1 or PD-L1 drugs which inhibit the immune checkpoint. So not surprisingly, gut microbes were found to be able to profoundly influence the potency of immunotherapy and some chemotherapies with immunostimulatory functions. And there is a growing consensus that the gut microbiome is linked to immunotherapy efficacy in the cancer patients. In fact, DNA sequencing of stool samples that are collected prior to checkpoint blockade therapy identified an association between gut microbiome composition and subsequent therapeutic response. Distinct bacterial taxa were overrepresented in the patients who responded well to the treatment, whereas other bacterial sequences or signatures were overrepresented in patients who are non-responders. And besides that, Bacteroides abundance was also found to be associated with good response to the ICB treatment, and a greater or more richer bacterial diversity was associated with higher response rates to the anti-PD-1 therapy. Therefore, with these revelations, there are several implications for future clinical applications and for research as well. Firstly, regarding the use of antibiotics in cancer patients who are receiving the ICB immune checkpoint therapy, clinicians should then review the use or abstain the use of or prescription of antibiotics. And secondly, microbiome could be used as a predictive biomarker for treatment outcomes, since higher bacterial diversity in the gut, but not the oral microbiome, was identified to be associated with better response rates to ICB. And thirdly, microbiota-associated therapies could be used to better improve immunotherapy treatment. Some of the microbiota-associated therapies are shown here. The less specific therapies include fecal microbiota transplant, which I will elaborate in a while, prebiotics like inulin fiber and proper diet, antibiotics to kill off some pathogenic bacteria, bacterial cocktail or consortia and probiotics, specific bacterial isolates or strains, bacteriophage to attack and kill off some of the undesirable bacteria. And the more precise method is, of course, the bacteriophage and also bacterial metabolites. A fascinating and yet a bit unsettling approach is fecal microbiota transplantation or FMT. So in this method, the donor species is collected from healthy donor or patients who responded well to the therapy, and then processed by mixing, blending, and filtration, and then transplanted either through endoscopy or in the form of pills orally to patients who are poor responder to therapy. So in fact, for it's been shown that for the anti-PD-1 immune checkpoint therapy, this FMT treatment actually helps. So don't be surprised that in the near future, if you are healthy and your gut microbiome shows a favorable composition, that you can even sell your feces and make money out of it. Yeah, just a joke. Okay, so I have started the talk with the question of whether microbes and cancer are considered as friends or foes. Now I'm going to end by saying that microbes and cancers are both considered friends and foes, depending on the situation and the exact microbes that you are talking about. So with that, I end my presentation. Thank you. Good afternoon.
All right. Now we thank you, Prof. Chong Pei Pei, for her presentation. I believe now we have a deeper understanding of the relationship between microbes and cancer. And participants, you are welcome to send your questions to Prof. Chong through this QR code, or you can click the Slido link sent in the Zoom chat box. All right, moving along. It's now my pleasure to introduce our second speaker, Dr. Lui Chong Ying. Dr. Lui is a senior lecturer at the Bio of Sciences, Faculty of Health and Medical Sciences in Taylor's University. He received his undergraduate training in biomedical science from University Putra, Malaysia, and further his master and PhD degree under the Japanese government scholarship in Tohoku University School of Medicine. After finishing his PhD, he worked as a postdoc at the Institute of Development, Aging, and Cancer in Japan. The main interest of Dr. Lui is drug design, development, and therapy. He was awarded numerous grants, including Fundamental Research Grant Scheme, as well as international grant from Taylor's University. He was also involved as a production scientist in Tessan Therapeutics in the world's first phase 3 immunotherapy clinical trial on nasopharyngeal cancer treatment prior to joining Taylor's University. Currently, Dr. Lui serves as an associate editor for BMC Complementary Medicine and Therapies, and he is also the editorial member of Cancer Treatment and Research Communication as well as Translational Oncology. Today, he will be sharing with us the topic from natural product-based drug discovery to cancer immunotherapy. Let's welcome Dr. Lui to deliver his speech. Welcome.
Thank you, Dolisha. Good afternoon, everyone, distinguished guests, participants, and the organizing committees. So today I will share my research experience on the natural product-based drug discovery to cancer immunotherapy. Next, please. So 7 years ago, we and a group of researchers from Boston published a research paper that described acting downregulation is the cause of one of the causes of Wiskott-Aldrich syndrome. So this F-actin assembly was disrupted, which causes the T cell unable to respond to the antigen. And this is one of the causes of the primary immunodeficiency in Wiskott-Aldrich syndrome. Next. So you'll be surprised that some of the patients, Wiskott-Aldrich syndrome patients, develop lymphoma, a type of blood cancer, and they have to undergo chemotherapy as young as 1 or 2 years old. Next, please. So what I learned from this study is, even though you have a sufficient number of immune cells, you have the T cells and B cells, but if they are not working, you are prone to develop cancers. Next. So when I came back to Malaysia, I was wondering, is there any natural product that can boost the immune system or act as a health supplement? Next, please. So we are focusing on one of the natural products here. As you can see, this is bitter cumin. It is also known as kaligiri or kaligeri in the Indian language. And the scientific name is Centrotirum enterminticum. Next, please. There are different varieties of cumin. I'm sure you're very familiar with the left and the middle one, the white cumin. The scientific name is Cuminum seminum. The black cumin, which is the Nigella sativa. And the rare type is the bitter cumin, which is the Centrotirum enterminticum. Next, please. So I'm sure you see this in the supermarket. This is on the right side from Babas. It's white cumin. In Bahasa, it's known as jintan putih or jintan manis, which we use to cook curry. On the left side is a black cumin, which you can buy from Lazada. The bitter cumin is the rare one, which you can only purchase from some medicinal farm. Next, please. Well, you can also buy it from Amazon, right? So you can see here, this is the packaging, and it is selling like 100 rupees, 179 rupees per 100 gram. Yep. Next, please. So Centrotrium entermeticum, or the bitter cumin, is widely used as a folk medicine for diabetes in Rayalaseema, India. So the Rayalaseema is a region comprised of Anantapur, Chittor, Kadapa, and Kunu in India. It is a very popular ingredient in Ayurvedic medicine, and in some other places, they are used as antihelmintic, diuretics, anti-phlegmatic, anti-asthmatic, and so on. Next, please. So our research interest is to evaluate the anti-cancer property of the bitter cumin seeds, and we try to find out the active compound that can exert this effect. And lastly, we try to understand what is the molecular mechanism of this active compound in cancer cells. Next, please. So first, we extract the compound using 3 different solvents: the hexane, chloroform, and the methanol. And we found that only chloroform extract shows significant cytotoxic effect towards cancer cell. So you can see here, when we stain with annexin 5, which is an apoptotic marker, only the chloroform extract responded to the treatment. Right. And on the right side, you can see from the flow cytometry, the JC1, which is the apoptotic marker, increases as we increase the concentration of the chloroform extract. So this shows that the active compound is inside this chloroform extract. Next, please. Next, we set to find out what are the active compounds that can exert this anti-cancer effect. Next, please. So we further perform high-performance liquid chromatography using the flash column C18, and we managed to get 6 different extracts. So as you can see from this chart, only the fraction number 5 and number 6 demonstrate significant cytotoxic effect towards the cancer cells, whereas the first 4 did not show any significant toxicity effect in the cancer cells. This shows that the active compound is contained in fraction 5 and 6. So we combine these 2 fractions and further perform isolation and ended up with 1 important compound. So to show the comparison, we also fractionate or recrystallize fraction number 4, and this yielded compound number 2. Next, please. So we perform LC-MS analysis and the MNR study using compound 1 and 2. And based on the structure analysis and the mass spec analysis, we identify that vernodellin is compound 1 in the previous slide. Can we go back to the previous slide? So this is the compound 1, 10 milligram, is vernodellin. And compound 2, it's a non-toxic compound known as the, next please, 12-13 dihydroxyolic acid. Next, please. So can vernodellin exert the same effects like the chloroform extract? Next. So we treated the breast cancer cells using vernodellin. So as you can see, Vinodolin inhibits the proliferation of both cancer cell lines in a dose-dependent manner and a time-dependent manner. Next, please. And when we performed Annexin 5 and PI study, we found that vinodolin able to induce apoptosis in a concentration-dependent manner. And you can see that from 3.125 microgram per ml, there's a significant increase of apoptotic cells. The numbers are increasing for the early and late apoptosis compared to the control. Next, please. And when we check the cell cycle, we found that vinodolin actually blocks cell cycle progression in the G0 and G1 phase. You can see that in the control, the G0 and G1 phase is around 64.8% in MCF7 and 55.4% in MDA-MB cells. Whereas in the treated cells, this fraction increased to around 72.5% in MCF7 and 61% in MDA-MB cells. So this shows that vinodolin is able to induce apoptosis and at the same time block cell cycle progression. Next. So vinodolin works very well in the breast cancer cell line. How about in an animal model? Next, please. So we induced a breast cancer using an animal model, and we treated the animal model using vinodolin. At the same time, we compare the treatment with tamoxifen, which is a standard drug for breast cancer. As you can see that the 10 milligram per kg of vinodolin is roughly equivalent to 1 milligram. The efficacy is roughly equivalent to 1 milligram per kg of tamoxifen. Whereas the efficacy of 1 milligram per kg is slightly lower, but it is also able to inhibit the breast cancer cell growth in an animal model. Next, please. Next, what is the molecular mechanism of this active compound in cancer cells? Next. So we did a few studies on some of the oncogenes and tumor suppressor proteins, and we found that vinodolin actually upregulates FOXO3A, which is the main target, and this in turn increases the Beam protein expression in the breast cancer cells. So you can see here in control, the cells are not expressing any FOXO3A, but when you treat it, and you can see there's a dose-dependent increase of FOXO3A in the treated cells. Right? So Beam is a downstream of FOXO3A. As you increase FOXO3A, you can see the level of Beam is also increasing. Next, please. So we are wondering, where is the location of FOXO3A? According to the publication, once FOXO3A is activated, it will translocate to the nucleus and activate Beam. So we did a nuclear cytosol fractionation, and we confirmed that the FOXO3A is located in the nucleus rather than the cytoplasm. This shows that the FOXO3A is being activated and it will translocate from cytoplasm to nucleus and activate Beam, which in turn activate the apoptotic process, which suppresses the tumor growth. Next. So how about the upstream? The upstream of FOXO3A is PI3K and AKT signaling. So we perform Western blot to check the expression of these two proteins. And you can see that the vinodolin is able to inhibit the phosphorylation of the PI3K or the AKT signaling, which is consistent with our observation that this in turn activates FOXO3A and causes the translocation. Next. So we mentioned previously that vinodolin is able to induce cell cycle arrest at the G0 and G1 cycle. And we have managed to prove that these two proteins are responsible for that particular effect. So the P21CYP1 and the P21KIP1 are the checkpoint proteins in the cell cycle. As you can see, when we treated the cells, these checkpoint protein expression are increased tremendously, which arrests the cell cycle into the G0, G1 phase. Next. So vinodolin is able to induce apoptosis through FOXO3A. What happens if we knock out FOXO3A? Next. So if we silence FOXO3A by using the siRNA, this will reverse the effect of vinodolin. As you can see from figure A, when we introduce siRNA, FOXO3A expression will be silenced. And at the same time, if you treat the cells with vinodolin, you will not able to see any increment of the checkpoint proteins, right? So this shows that the effect of vinodolin requires FOXO3A as a mediator to induce apoptosis in breast cancer cells. Next. So we were able to recapture this phenomena in the tumor tissue as well, right? Not just on cell line, but we're able to see an increment of FOXO3A and P27KIP1 in the treated tumor tissue. Next. So in summary, these bitter cumin seeds contain an active compound known as the vinodolin. However, as I've shown you, the efficacy of this vinodolin is 10 times lower compared to tamoxifen. So that means we need to do a lot of improvement before we can move it into the clinical phases. And unfortunately, this vinodolin has a direct effect on the tumor tissue. So the number of T and B cells in the treated animals were not much different. Right. So we are not able to see any differences in terms of the immune system of these treated animals. Next, please. So we have published this in the journal. Next. The animal studies, if you're interested, you can have a look on this journal as well. Next. At the same time, there's a shift in the from chemotherapy to a very groundbreaking technique known as immunotherapy. In 2014, if I'm not mistaken, there are several landmark papers that are published in Science and Nature describing a breakthrough using your own immune system to fight cancers. Next. As you know, in 2018, 2 cancer immunologists were bestowed with the Nobel Prize for their discoveries of PD-1 and CTLA-4 by Professor James Allison and Professor Tasuku Honjo. Next. So in order to know how we can perform immunotherapy in patients, so I previously joined this company known as Tessa Therapeutics. Next, please. So this is one of their pipeline. This treatment is known as a CD30 CAR T cell therapy, which is still in clinical trial. Next, please. So what is CAR? CAR is a short form for chimeric antigen receptor. It is an artificial T cell receptor that have been engineered for cancer therapy. So in the middle, you can see this figure. This is an engineered T cell receptor. So it has a FV region that can recognize cancer antigen. So on the right side, once you genetically engineer these T cells to express these CAR receptors, these chimeric receptors, it will start to recognize cancer antigens. And once it binds to the cancer antigens, it will kill the cancer cells. So it is very specific, right? The T cells will now start to attack and kill the cancer cells. Next, please. So what is the process used in CAR T cell therapy? First, the T cells are collected from the patient blood and they are used to produce a special receptor on the surface, which is the chimeric antigen receptor. And in the lab, the T cells will be grown in sufficient number so that it can be infused back to the patient. Next. So in 2017, the US FDA approved 2 of the CAR T cell therapies, namely the Kymriah, the first CAR T cell therapy, and the Yescarta. Next. So the reason why we are not using Kymriah or Yescarta technology is because these 2 therapies basically target the CD19, positive hematological malignancies. CD19 expressed in both B cells, malignant B cells and normal B cells. Therefore, Kymriah and Yescarta can kill both normal and cancer B cells because CD19 is expressed in both fractions. But CD30, on the other hand, is only expressed in Reed-Steinberg RS cells in Hodgkin lymphoma. So using these specific antigens, you can specifically target the Reed-Steinberg cells by the T cells, so you don't affect the other B cells. Right, next. But there are many challenges before we can use immunotherapy in real life. For example, the cost. The cost of CAR T cell therapy is equivalent to like buying a Lamborghini or Mercedes-Benz S-Class, right? So the second thing is you need a infrastructure and a very well-trained personnel. And this infrastructure must be GMP compliant, right? And the dosage, how many cells that should be infused into the patient, right? Because there are certain side effects, right? Some patients develop side effects, for example, cytokine storm, right? So these issues have to be overcome before the use of immunotherapy. Next. Okay, that's all for my presentation. Thank you very much. Again, I thank the organizing committee for the invitation for this presentation. Back to you, Alicia.
No problem. Thank you, Dr. Lui, for the sharing. We have learned so much from you today. Ladies and gentlemen, feel free to upload your questions for Dr. Lui through Slido. So next, I'd like to introduce our third speaker, Prof. Dr. Ho Guo Huang. Prof. Dr. Ho is an associate professor and clinical oncologist, radiotherapist at the Medical Center and Specialist Center in Kuala Lumpur. He was trained at Barts and the London National Health Service. He attained his certificate for completion of specialist training in 2007 and joined the Faculty of Medicine at He was the recipient of the Joint Commission International Outstanding Young Malaysian Award in 2009 for medical innovation. He's the oncology lead of the Center for Image-Guided and Minimally Therapy Stereotactic Radiosurgery Project at as well as sub-investigators for high-impact research grant projects. His research interests involve breast, gastrointestinal, and gynecological cancers. He's involved in many national and international collaborative research work. Being a council member of Together Against Cancer, he champions cancer patients' rights in Malaysia. It is now my pleasure to invite Prof. Dr. Ho to share with us on a topic entitled Cancer Care Through COVID-19 and Beyond. Welcome, Prof.
Thank you. Hi, good afternoon, everybody. I have 20 minutes to talk about cancer care, which is a very big topic. I hope I can introduce you some idea to the concept of different aspects of cancer care. this is the world cancer map. As you can see, the countries with the highest incidence of cancer actually are the developed countries. So we don't know what is the reason, but this is a pattern that we see. So one would feel that this is not just to do with the genes, but also to do with some environmental factors. In Malaysia, breast cancer is the commonest cancer, followed by colorectal and lung cancer. In men, colorectal cancer, lung cancer, prostate cancer, nasopharynx cancer, and liver cancer are the top 5 cancers. In women, breast cancer, colorectal cancer, cervix, lung cancer, and ovarian cancer. What we do know is that the cancer incidence is rising, and so is cancer mortality. Breast cancer mortality will be rising 43% from 2015 to 2030. Now, the big question, of course, is what causes cancer, which a lot of the causes are actually unknown. This paper in Science 6 years ago caused a huge uproar by claiming that most of the cancer is just due to bad luck, i.e. cell division causing mutations and then leading up to cancer. However, subsequently, the paper in Nature told us that there is substantial contribution of extrinsic risk factors to cancer development. Actually, probably only 10 to 30% of cancers are due to unavoidable intrinsic risk factors. So now it's commonly accepted that about 40% of cancers can be prevented. These are the cancers, the lifestyle changes that we can make to prevent cancer. A lot of them: keep a healthy weight, drink less alcohol, eat a high fiber diet, cut down on processed meat, breastfeed, and be more physically active. Actually are something that one can do every day. However, that brings up the question of preventive medicine. So the best doctors prevent sicknesses. So that's actually what one should be talking about when we talk about cancer management. The modern illnesses are due to a lot of aspects of modern lifestyle: lack of physical activity, lots of junk food, a lot of food industry pushing different food product beverages to us. Obesity is a major issue in our country. Just have a look in the hospital. You can see a lot of obese staff and patients. Too much meat, too much animal fat, and not enough vegetables and fibers. So the proportion should be reversed. Much more vegetables and much less meat product. Why? There are many studies that show that plant-based diet prevent cancers. Most of the studies are done in common cancers like breast cancer. For example, carotenoid prevent breast cancer. Photocarotenoid cut down the risk of breast cancer by 19%. Flavonoids or flavonoid subclasses also reduce breast cancer risk, in particular flavone and flavonols. So plant-based diet contains a lot of plant alkaloids, which are very good for our health, like beta-carotene, lutein, resveratrol, carotenoids, isothiocyanates, we just heard curcumins, catechin, and a lot of beans. So the Women Intervention Nutrition Study basically set out to change people's dietary habit by reducing dietary fat, mainly animal fat, and found reduce in relapses at 7 years for those who managed to change their lifestyle. And more remarkably, it's not just in ER-positive breast cancer patients, but also in ER-negative breast cancer patients. So many studies show that physical activity reduces mortality risk for cancer patients. Of course, there's a limit how much reduction you can get if you exercise, if you walk 1 hour a day for 7 days a week, you can cut down the risk of cancer death by 39%, or run half an hour a day for 5 days a week. But the ordinary doctors or mediocre doctors like us talk about illnesses. So we will spend the rest of the talk on illnesses. When you talk about treatment of cancer, you also talk about, you have to divide them into early disease or late disease. That is important so that you use the right approach. So for early disease, where the cancer is localized, you use a local regional approach. For late disease, when cancer has spread, you use a systemic approach. Local regional approach is very effective for a localized tumor. High success rate, one-off procedure. So you don't want to use a systemic approach for a localized disease. That would be a wrong approach and you will have low success rate, and vice versa. So late disease, where cancer has spread to many sites, use systemic approach. You do get success sometime, but the success rate is much lower than local regional approach for localized disease. And this one requires long-term management. For example, so localized disease treated with local regional approach, like surgery, most important treatment for localized cancer, head and neck cancer, lung, GI tract, urological, gynecological, or CNS cancers. The surgery is still the most important modality if the cancer is localized. Different types of surgery like robotics, Da Vinci robot, using laparoscopic method, liver resection. Last time when patient has liver metastasis is considered end of life, but now let's say for colorectal cancer, liver metastasis, you can still take out 1, 2, 3 segments and patient can still survive. Success rate of 30% long-term survival. And then there's image-guided surgery, doing a scan during surgery, particularly for brain surgery. Second main modality is radiotherapy. And then you have different types of radiotherapy: conventional radiotherapy, intensity modulated radiotherapy, image-guided radiotherapy, the stereotactic technique, intraoperative technique, brachytherapy or implant. And then there is particle irradiation like proton or carbon ion that we don't have in Malaysia. So this is intensity modulated radiotherapy, IMRT, to treat nasopharynx cancer. So you give it high dose to the PNS without damaging the throat or the mouth. IMRT for a rib metastasis, that you have to spare the spinal cord. Stereotactic radiosurgery using a cyberknife. So from multi angles, you irradiate the tumor to achieve a very high dose at the center. Intraoperative radiotherapy, like this TAGIC. So radiotherapy reduces local recurrences and increases breast cancer survival. So breast conserving surgery, relapse rate 30%, breast conserving surgery and radiotherapy, relapse rates 10%. Mortality rate also lowered with radiotherapy. This is a patient with lymphoma. Halfway through radiotherapy, the tumor has dissolved. So other local regional approach like cryoablation use extreme coldness to freeze the tumor and kill it, or radiofrequency ablation using heat. Radiofrequency waves generate heat and basically kill the tumor in the body. Selective intrahepatic radiotherapy, insert radioactive yttrium through the groin, get into the arteries, into the hepatic artery, and inject this into the tumor and irradiate it from inside. This is the tumor before. So we call that SIRT, selective intrahepatic radiotherapy. This is after. Systemic approach, the most well-known one, of course, is chemotherapy. But chemotherapy does not cure many patients. Many patients with germ cell tumor, lymphoma, or leukemia. Otherwise, it's used for adjuvant treatment or palliative treatment. This is a patient with metastatic NPC. He had palliative chemo that worked very well. So now we talk about personalized medicine, because if you take 100 patients and these are the genes that you can analyze, if the genes are active, the genes are not active, you can actually plot everybody's gene profile in this diagram, and you can classify maybe 2 or 3 types of different tumors, which depends on their gene expression profile. So we know there are 10 characteristics of cancer cells: evading group suppressors, sustaining proliferative signals, avoiding immune destruction, enabling replicative immortality, tumor-promoting inflammation, activating invasion and metastasis, forming blood vessels, unstable genes, resist cell death, and dysregulating cellular energetics. So you can target all different aspects of the cancer. For one example, EGFR mutated lung cancer treated with EGFR inhibitor, the tumor basically gets smaller after 1 month. ROS1 mutated lung cancer treated with crizotinib, ROS1 inhibitor, also works very well. The lungs totally clear up after crizotinib. This is chemotherapy and anti-EGFR antibody for colorectal cancer with liver metastasis. Totally we talk about immunotherapy. So that is the latest development in oncological treatment. So we're talking about the engagement between the dendritic cells, antigen presenting cells, that activate the T cells that go and attack the tumor. So that is the process. So we heard just now that checkpoint inhibitor, for example, nivolumab, has changed the treatment landscape of lung cancer, for example. So this is 2 years out, 20% of patients still survive, better survival rate than chemotherapy, docetaxel. This is pembrolizumab versus chemotherapy. This follow-up is for 5 years. You still see that 31% of patients are alive at 5 years for advanced lung cancer, double that of chemotherapy or targeted therapy. So this basically changed the treatment protocol. This is one of our patients who had a new checkpoint inhibitor called camiplimab in a study. So June 2018, huge mass in the right upper lobe. October 2019, totally dissolved away. So very impressive result. This is another patient, bigger mass with mediastinal lymph node, much smaller after 1 year of treatment. This is a third patient, huge mass in the left lobe with pleural effusion, disappeared after 1 year of treatment. So we see quite a few of these patients responded really well to immunotherapy. This is my first patient treated with immunotherapy. She had advanced gastric cancer that grew out of her umbilicus into the skin. We call it Sister Mary Joseph nodule, and it's bleeding. So she had pembrolizumab, and then after a few months, the tumor just disappeared. That was 5 years ago now. So she's essentially cured of advanced gastric cancer, never heard of in the past. But now we know that checkpoint inhibitors only work for essentially 10 to 20% of patients, not all cancer. These are the main cancers that we can use it for. With the exception of melanoma, the response rate got up to 40%. So lung cancer, kidney cancer, bladder cancer, liver cancer, ovarian, this one not so effective. Gastric, esophageal, head and neck, and triple negative breast cancer. Response rate is around 15 to 20%. That's why my colleague, Professor Teo Su Huang at Cancer Research Malaysia, they have done a lot of basic work on the genomic profile of breast cancer patients in the country. And they came up with an idea that a lot of Malaysians actually carry ApoBac3B deletion, much more common than the Westerners, 37% in East Asians versus 6% in Caucasians. And these kind of mutations basically make the tumor more immunogenic, a lot more immune cells and rich immune score in the Asian breast cancers. So, together we designed a study called Phase 2 Single-Arm Open-Label Pembrolizumab in Metastatic HER2 Negative Breast Cancer Patients called ORAL study. That was named by me. One good thing of practicing medicine is that if you design a trial, you get to name it. So the next phase, I think my personal feeling would be combination immunotherapy. All the studies are ongoing now. And cell therapy, Dr. Louis talked about CAR T cell just now. There are tumor infiltrating lymphocytes and cancer vaccine. And gene therapy, like using antisense oligonucleotides, microRNA inhibitor, or you just change the gene of the patients with CRISPR technology. But after a couple of decades, then we'll be in the era of nanomedicines, because the nanorobots using DNA or RNA would be used to basically kill all the tumor and correct our genes inside our body. So nanomedicines will be the next major breakthrough, probably another decade to come, and also AI in medicines. But I still feel that doesn't matter how effective or how advanced is the modern technology, prevention is still better than cure. Prevention is better than cure. That should be the motto. And you are what you eat, because that's what happened. So you should take plant-based diet of different colors and reduce and more fibers and reduce animal product or animal fat. Thank you.
Thank you, Prof. Dr. Ho, for the great presentation. Ladies and gentlemen, you can type your questions on Slido, as our speakers will be answering your questions in the open forum later. Next, for participants collecting CPD points, you can scan this code with your MMA app to self-register your attendance within 30 seconds. For those who have not scanned, don't worry, as you'll be receiving the points within the next 7 days if you have registered. We will also be sharing the code again at the end of this webinar. Thank you. So later, we will invite Prof. Wang for the next presentation. Okay, so moving on to the next slide. I'd like to introduce our fourth speaker, Prof. Dr. Yu Ming Wang. Prof. Dr. Wang is currently the chairman of the Department of Radiation Oncology, Proton and Radiation Therapy Center in Taiwan, Kaohsiung Changkeng Memorial Hospital. He received his undergraduate training in Taiwan, National Yang-Ming University. After graduating, he worked as a resident and later an attending physician in radiation oncology, Kaohsiung Hospital. Recently, in the 24th Taiwan Joint Cancer Conference, he was the runner-up for the Best Clinical Research Award. Today, he will be sharing with us on the topic, Treating Cancer with Proton Beam Therapy, experience from Kaohsiung Changkeng Memorial Hospital, Taiwan. Let's welcome Prof. Dr. Wang to give his talk. Welcome, Prof.
Good afternoon, ladies and gentlemen. It is my great honor to have this chance to share my clinical experience with proton beam therapy with you. My name is Yu Ming Wang, Wang Youming 医师, and I'm currently the chairman at our department and the Proton Center. Today's presentation, I would like to divide it into mainly two parts. The first one is basics of proton beam therapy, and the second one is clinical experience at my institution, Kaohsiung Changkeng Memorial Hospital. Okay, let's go to the first part. Before we talk about therapy, we need to know first what is a proton. A proton is a positive charged particle in the nucleus of an atom. For clinical PBT, we use hydrogen to generate protons. Before we talk about therapy, we need to after protons are generated, then we put those protons into a cyclotron. The cyclotron will speed those protons up to the speed around 70% of the light speed. Then, those high-speed protons will be transferred through the beamline to the gantries, and then delivers to the human body. Those high-speed protons can kill cancer cells, just like the high-speed small rock tied on the sling swung by King David can kill giant Goliath. But how can those high-speed protons kill cancer cells? Protons hit the DNA double strands and break it, so the cancer cells will lose their ability to grow and survive. After we know how protons can kill cancer cells, another thing we need to learn is the characteristic of proton. This picture shows the relationship between the dose and the distance after a proton goes into the body. We can see that protons will deposit most of its dose at a certain depth, while giving only a little of its dose before it and zero dose after it. This is what we call Bragg peak. How do we control the Bragg peaks? Actually, protons differ with the highest speed, which deposit its dose more deep, while protons with lowest speed, just like the blue line, will deposit its dose more close to the surface. Why do we need to control the depth of Bragg peaks? Because cancer cells always have its size. They won't be like a tiny spot inside the body, but have some thickness, just like 2 centimeters or 3 centimeters in size. So when we need to kill cancer cells, we need to generate an effective tumoricidal dose with thickness. By controlling the distribution of Bragg peaks with different energies, we generate a spread out of Bragg peaks, or SOBP, for clinical use. After we know those basics of PBT, then we need to learn what is the difference between PBT and photon therapy, or we say X-ray therapy. The biggest difference is the dose distribution inside the body. A proton will give its dose just like the blue color line, as what we have learned. But a photon, or X-ray, or conventional radiotherapy goes differently. Photon gives it most of its dose, like the green line, highest near the surface of the body and penetrates infinitely through the whole body. Why the different dose distribution inside the body matters? Because the dose not only kills the cancer cells, but also damage the surrounding normal tissues. This picture is a comparison between proton beam therapy versus photon radiotherapy. If a tumor, the gray part of the picture, located at 19 centimeters to 26 centimeters inside the body, which is quite large, the different dose distribution will go like this. For proton therapy, the high dose parts will be just at the tumor, while tissues before and after the tumor receives low dose or even zero dose. But for photon, it's not like this. Just like the yellow line, photons tend to give high dose before the tumor and hurt tissues after the tumor. Those unnecessary doses will cause unnecessary complications, or we say side effects. So that is why proton therapy always have lower side effects comparing to photon radiotherapy, while the tumor control rate won't be affected. This table gives a brief comparison between clinical use of proton therapy and photon radiotherapy. The dose distribution is the key factor that makes the large difference. However, PBT centers are not like photon radiotherapy centers. There are currently only 2 PBT centers in Southeastern Asia, and all of them are in Taiwan. Besides, the technique and clinical experience to control proton beam therapy is much higher and complicated. Okay, now a brief summary of the first part. We learned what is PBT, how PBT works, and why PBT is better. Okay, let's go to the second part, clinical experience. Let me introduce my institution first. Kaohsiung Changgeng Memorial Hospital is a tertiary referral medical center in southern Taiwan and was built in 1986. There are 4 buildings in our main campus and over 2,600 beds. For cancer treatment, more than 6,000 newly diagnosed cancer patients will have their treatment in our institution every year. Our proton beam therapy center was started in operation since late 2018. In the past 2 years, more than 600 patients have received PBT treatment in our center. 1/3 of the patients were head and neck cancers, and 1/3 of them were hepatocellular carcinomas. So, in the following presentation, I would like to share more detail about some specific disease with PBT results. One is nasopharyngeal carcinoma and head and neck cancer. As we know that traditional photon radiotherapy has already given very good tumor control to those cancers. But the most disturbing part of the treatment comes from toxicities, or we say side effects. The benefit of proton is the reduced side effects. What are some common side effects of photon radiotherapy when treating nasopharyngeal carcinoma or head and neck cancers? Common toxicities during treatment include mucositis, dry mouth, and taste change. After treatment completion, patients tended to have dental and hearing problems, dry mouth, as well as soft tissue fibrosis. The most frightening part for patients receiving head and neck region radiotherapy is mucositis, which is ulcer, erosion, and defects of the oral mucosa caused by radiation damage. In the clinics, we defined gradings of mucositis according to its severity. Patients with grade 0 to 1 usually have no clinical symptoms and won't affect their normal daily life. Patients with grade 2 have some ulcers, but most of them can still tolerate oral intake. However, for patients with grade 3 or higher mucositis, tube feeding is always mandatory for nutrition and fluid supply, and patients always have severe pain causing them fear to following treatments. Grade 2 mucositis is like this: pain but still tolerable. Of course, we like to avoid it. For grade 3, it's like this. You can see the erosion over the mucosa, and you can easily imagine the pain and fear for this patient. In addition to the mucositis, long-term and late dental problem is another issue that has high impact on quality of life. Due to the radiation exposure during radiotherapy course, dental caries, periodontitis, and even severe osteoradial necrosis can develop years after treatment. This picture is a typical look years after radiotherapy. Nasopharyngeal carcinoma is a tumor located at the central part of head neck region. Just like the red circle on this picture, nasopharynx lies deep inside the nose and just in front of the brain. The benefit of treating nasopharyngeal carcinoma by using proton beam therapy can be easily understood in this cartoon. Look at the left side of the picture. If we treat NPC with photon radiotherapy, the dose would be higher at the nose, the mouth, as well as the brain. With PBT, the dose hits the tumor effectively, with very low dose to the surroundings. The same idea applies to the right picture. Dose to the ears, cheek, and parotid glands tends to be higher for photon radiotherapy. In results and in my practice, patients with NPC treated by proton beam therapy would have their dose to those parts reduced significantly. 60% lower to the ears, 80% lower to the glottis, pharynx, and esophagus, and almost 100% lower to the teeth and some parts of the oral cavity. A brief result of our current practice. In the past years, we have treated 49 NPC patients with proton beam therapy in curative intent. Most of these patients have advanced stages. For the side effects, we can see that around 90% of patients had only grade 0 to 1 mucositis. Comparing to our own historical data with photon radiotherapy, more than 70% of patients had grade 2 or higher oral mucositis. What about the tube feeding rate? Historically, it was around 10 to 20%. However, amazingly, tube feeding rate is 0 in my practice. The 2-year overall survival was 97%, and only 5% of patients had their disease in recurrence, which I think is quite good. In addition to NPC, we have also treated a lot of advanced cancers arising from the head neck region, like this 41-year-old woman, an artist, with left side sinonasal cancer with incomplete surgical resection. After treatment, she has very minimal side effects during the course, and the tumor is in complete control 2 years after high-dose proton beam therapy. Media have reported her story here in Taiwan. Another one is a 56-year-old teacher who has recurrent huge left ear cancer invasion to his brain. Surgeons had told him that the disease is not able to be cured by surgery and referred him to my clinic. I prescribed very high-dose proton beam therapy to the tumor. Thanks to the bright peaks of proton beam therapy, although the dose was very high at the tumor, there were only very minimal dose to the surroundings. He had no mucositis and other side effects during the treatment, except grade 2 dermatitis. 1.5 years after proton beam therapy, we can say that his disease is cured. He is now back to the school teaching his students. His surgeon was amazed by these results. After nasopharyngeal carcinoma and head neck cancers, let's talk about hepatocellular carcinomas. HCC located at the central part of the abdomen. If we want to treat HCCs by conventional photon radiotherapy, the dose would be high at surrounding normal liver, and the dose to the gastrointestinal tract would also be high because photons won't stop at the tumor. However, with PBT, the dose can be precisely delivered to the tumor without damaging normal liver and guts. With PBT, we are able to decrease normal liver dose by 50% and increase the dose by 30 to 50% comparing to the era of photon radiotherapy. We have published our results with PBT to HCCs. Our current data reveal that in-field local control was around 100%, and the side effects called radiation-induced liver disease was only 5%, which was so low comparing to our own historical data. Another disease I would like to mention is esophageal cancer. Since it is at the central part of the thorax. Conventional photon radiotherapy would cause high dose to the heart, the spinal cord, and the lungs. With PBT, the dose would be lower. Our own experience showed that the lung dose was decreased by 80% and the heart dose was decreased by 60%, so that it preserved the lung and heart function for patients who is going to receive following surgical resection. For patients who declined surgical resection, we are currently conducting a clinical trial using higher dose to the tumor, which theoretically would increase the local control. Comparing our limited experience with high-dose proton beam therapy for esophageal cancer to our own published historical data, the tumor control rate was excellent. 3 out of 3 has their disease reached clinical complete response. And we are looking forward to more patient accrual and the long-term results. With the limited time, I'm not able to report more of our clinical results. However, in summary, our patients and us did experience the theoretical benefit from BRAC peaks, a game changer when fighting with cancers. COVID-19 has changed our daily practice a lot. Do we have preparation for that? Yes. As a big medical center with such modern modality, now patients treated at our center are asked to wear masks during their course of treatment. Will that cause some influence for the dose of proton beam therapy, since the BRAC peaks are so sensitive to the depth, especially for head-neck region? We have published our own results demonstrating how to adjust our protocol during this pandemic. We all hope COVID-19 will disappear soon, but wishing for the best and preparing for the worst is always our responsibility. Thanks to your attention, and this is the end of my presentation. Thank you.
Thank you, Prof. Dr. Wang, for the informative sharing. If anyone has questions for Prof. Dr. Wang, kindly send them through this Slido link. Thank you. Now, I'd like to introduce our last speaker, Dr. Sound Cherry. With over 20 years experience in cancer control, Dr. Sound Cherry advocates for the awareness, prevention, early detection, and survivorship of cancer. Within Malaysia, these initiatives are driven through the National Cancer Society of Malaysia, the nation's very first cancer NGO. Dr. Sound Cherry studied medicine in Dublin, Ireland, and has an MBA from the University of Bath. Locally, she's also on the board of Secretary of the Malaysian Women's Action Tobacco Control and Health and the Malaysian Council for Tobacco Control. The accumulative of clinical, educational, and counseling skills has provided her a comprehensive picture of the benefits and challenges of cancer control in Asia. In November 2020, she was conferred the Darjah Datuk Paduka Mahkota Perak Award, carrying the title Datuk by His Royal Highness, the Sultan of Perak. Without further ado, I'd like to invite Dr. Sound Cherry for her presentation on Sealing Cancer Survivorship. Welcome, Dr. Sound Cherry.
Good afternoon, everyone. Thank you for having the National Cancer Society of Malaysia and myself at this webinar today. Sailing Cancer Survivorship. Cancer management is not just about the diagnosis and treatment of cancer, but it encompasses all the steps required to ensure the best possible survivorship journey. Hence the title, Sailing Cancer Survivorship. So in the next 20 minutes, I'd just like to talk a little bit about how NCSM contributes to sailing cancer survivorship in Malaysia. The services of NCSM are divided into 3 pillars: educate, care, and support. Each pillar having key areas of interest. In the next few slides, I'll go through these 3 pillars and how this impacts on cancer care in Malaysia. So the first pillar is educate. And as you can see, educate has been divided into 4 subsets. The first being health education, the second literacy, the third promotion, and the fourth policy. In health education, we focus primarily on the prevention and early detection of cancer. And this is primarily to the general public, but it also encompasses healthcare professionals. In health literacy, it's very much the skill-based workshops, community screenings, and medical camps. And this really empowers individuals to take control over their health. With promotion, one of the things we do is the school-based smoking cessation program. And this is an intervention program to assist people in reducing their health risk of developing cancer. And the fourth, which is the health policy, it is to empower Malaysia to enact change within the healthcare. And that means empowering government as well as community to enact change. For health education, as you can see here, NCSM produces various resources. These resources can be cancer specific or on general risk factors of cancer, and they're in various languages. As you can see on this slide, there's a leaflet on the HPV vaccine, screening tests for colorectal cancers, the type of things that you need to look out for in colorectal cancers, and talking a little bit about sugar, which increases the risk factor of obesity, which is linked to cancer. The Cancer and I sessions are sessions which go over a few months, and they're awareness sessions for the general public. Again, they can be on specific cancers or on general topics. The first one you see there, especially for the gentlemen, are on cancers which are specific to men, whilst before and after, talks about adjuvant and neoadjuvant therapy, so the type of treatments available generally for cancer. And the third you see is again on specific cancers, colorectal cancers and ovarian cancers. So whilst these Cancer and I sessions are curated by the National Cancer Society of Malaysia, we work in collaboration with different hospitals. As you can see below, that we've worked with Prince Court, with KPJ and Pantai Hospitals, and many of the other private hospitals. And though they are specifically for the general public, they also do benefit healthcare professionals, especially those in the primary healthcare setting. So we're talking about GPs, the frontliners who see cancer patients on a daily basis. We don't only focus on the medical aspects of cancer. So the Fight, Thrive and Survive series is a series of education sessions for cancer survivors via Facebook Live. And these can go through various topics. And what I've shown you here are topics on psychosocial support, on nutrition and diet for children with cancer. So these series are all in different languages. So it's in Malay, English, as well as Chinese. The work we do on health policy is to strengthen and elevate our health systems in Malaysia. And National Cancer Society of Malaysia doesn't do it alone. We have many partners that we collaborate with, be it universities, other cancer organizations, political parties, government, as well as the Ministry of Health, of course. Our work in health policy took to center stage in 2018 when the NCSM hosted an international congress with UICC, the World Cancer Congress. And this was a huge feat for Malaysia because it really put cancer in the forefront of health in 2018. And to be honest, it wasn't just Malaysia, but it was a huge feat for the whole of Southeast Asia. And the areas which were discussed were in cancer control as well as in tobacco control. And in cancer control, we looked at big picture issues such as the National Cancer Control Program or the ASEAN Cancer Resolution. But we also look at specific challenges which are facing Malaysia, such as the public-private referrals for patients with cancer. And tobacco control is a huge area which impacts greatly on risk factors in terms of cancer. And not just cancer, but in non-communicable diseases in total. So there were insights into the Framework of Convention for Tobacco Control, the FCTC, the Tobacco Control Act, and we pushed at that time for smoke-free eateries, which came into being last year. NCSM has a team that works tirelessly on policy briefs and reports which are specific to cancer, looking into the early detection and screening of cancer, the impact of biosimilars in the country, universal health care in the Malaysian cancer context, and things like reintegrating children who have had cancer back into schools. So they're big picture issues as well as honing in on specific issues, just like getting kids back into school. But we cannot just focus on cancer. We need not to just look at cancer in a silo, but we need to look at cancer in the bigger picture of the whole health system. So that means that we need to look at other diseases which have commonalities with cancer so that we can optimize, synergize pathways for better care for patients in general. And that's why the National Cancer Society of Malaysia gave being to NCD Malaysia, another organization which looks at non-communicable diseases as a whole. So this looks at cancer as well as heart disease, as well as mental issues, as well as lung disease, all which have common risk factors and maybe common pathways in terms of treatment and post-treatment care. So by having an organization such as NCD Malaysia, we can emphasize the patient's voices so that policies are not made from top down, but also encompasses the views and voices of patients. It also ensures there's capacity building for patient groups. So the actual setting up of patient groups, but also empowering patients in taking charge of their health. And it looks at policies and gaps among healthcare professionals in managing NCDs and the universal health coverage. And as I mentioned before, NCSM doesn't do these things alone. For us, it's really important for us to work with other organizations who are like-minded, who have similar values, similar philosophies, and similar goals in terms of cancer as well as NCDs in Malaysia. So we work on an international level with the NCD Alliance, UICC, Childhood Cancer International, ICISG, as just a few of those that we work with, WHO as well. And we also work with organizations within the Malaysian context. So the Malaysian Medical Association, Malaysian Council for Tobacco Control, MyWatch, which is Malaysian Women's Action for Tobacco Control and Health. And these are just a few of the partners that we work with. Because only if we work together can our voice be strong and we can make a huge difference to healthcare in Malaysia. So the second pillar of the National Cancer Society of Malaysia is the care pillar. And here we actually have standalone clinics, the Cancer Health Screening Clinic, the Nuclear Medicine Center, and we also do community projects. The Cancer Health and Screening Clinic is a one-stop screening and diagnostic center that provides health and cancer screening for men and women of all ages. So basically, it makes diagnosis more accessible to all Malaysians in the more common cancers such as breast, cervical, and colorectal cancers. And these services are subsidized or provided free. So some of the services that we've listed here are the digital mammography services, ultrasound for the breast, thyroid, abdomen, pelvis. We do ultrasound-guided breast biopsies as well. Pap smears for cervical cancers, vaccinations, and your basic x-rays and ECGs as well. The Nuclear Medicine Center is slightly different. It's a center which provides diagnostic services. So usually someone already has cancer and needs to know where the cancer has spread to other organs or bones, so that the doctor has a better idea of the cancer extent before initiating whatever treatment which is required, be it surgery, radiotherapy, or chemotherapy. So this is a referral center for other hospitals, both from the public or private sectors. And here we provide bone scans, renal scans, as well as thyroid scans. The community projects that NCSM does are different in the sense that the previous two slides I showed you were our in-house clinics. This is where we go out into the community. And here we provide basic screening at the community level, especially for the underprivileged community. And it's important to note here that the team that goes out are just not healthcare professionals. It's a multidisciplinary team which consists of healthcare professionals as well as survivors. And that's really important because the voices of survivors are louder in the community setting, and they resonate with people, and it encourages people to actually empower themselves in taking control over their health. And of course, a huge part of our team are the volunteers who help us out as well. This is one of the community programs that we have. It's the Etika Mammogram Screening Program. So this has been ongoing since 2017, and we're starting on our 4th year this weekend. And it works with 14 private hospitals around Malaysia to provide the screening service. It's available for Malaysian women over the age of 40 with a household income of 5,000 ringgit and below. So in the 4 years, we have done 17,000 free mammogram screenings and 3,000 free pap smear screenings over 10 states. It is the largest non-governmental run breast cancer screening initiative in the country. This is the leaflet explaining what it's all about and how to actually enlist onto the program. And these are the partners and the areas that we've worked in. So this is one of the largest community programs that NCSM does on breast cancer, which is the #1 cancer in Malaysia. The third and final pillar of NCSM is our support pillar. NCSM, I have to say, strives to be by the side of a cancer survivor throughout their whole journey. So we do this with the mechanisms here that we have in hand, which is the Resource and Wellness Center, the psychosocial support network we have, our Cancer Information Service, and our Children's Home of Hope. So the Resource and Wellness Center was started in 2002, end of 2002, and it's a center offering services centered on supporting those affected by cancer. And when we talk about those affected by cancer, we're not talking just about the cancer patient, but you're talking about the family, you're talking about the caregiver who looks after that person with cancer. So it's really just not looking after one person, but looking after a whole family. And the focus of the Resource and Wellness Center is on the wellness of the person and their surrounding support system for recovering from the disease. It's all complementary. And of course, it's a big part of the fundraising efforts of NCSM so that we can sustain the Resource and Wellness Center and our psychosocial support. So the activities that we have are numerous. A lot of them are on wellness classes. So wellness classes can be yoga, it can be cooking, it could be Chinese ink painting, dancing, etc. There's talks as well, which are medical and health related issues. And there's survivorship masterclasses. So it's really focusing on the person with cancer, making sure that cancer doesn't overwhelm them, that they don't lose their identity to cancer. I think it's very important to note that when a person gets cancer, it's immediately, that lady has breast cancer. So a person loses that identity straight away. If a person gets heart disease, it's not that you introduce someone to say, that lady has heart disease. But cancer has that persona about it. And because it does, that person who does have cancer loses that identity and sense of purpose. And so the Survivorship Masterclass is really empowering people back to realize that it is them as a person who has a disease, not a disease which has overtaken them. And so it's very important for people to realize that a cancer survivor is a person with a disease and not vice versa. And we help out in things like this as well with the support group. We have amazing support groups in NCSM. The Women's Support Group, which is Pink Unity, the Young Adult Support Group, the Male Support Group as well. So there's a lot of activities surrounding survivorship in National Cancer Society of Malaysia. With psychosocial support, again, there's many activities that we do, and I'll just quickly go through them for you. The 4 components of the psychosocial support system that we have at NCSM is the play therapy service, which is provided by a play therapist, and it's delivered through our Children's Home of Hope, which I'll talk about to you later. The second is a psychological service provided by a clinical psychologist, the counseling service provided by a counselor, and diet consultation service provided by a dietitian. So all these are issues that impact a cancer patient outside of their purely medical aspect of their cancer. And these services are delivered through our Cancer Information System, or what we call our Cancer Toll-Free Helpline. Our CIS, our Cancer Information Service, provides cancer support information. So the target group are cancer patients, their families and friends, even healthcare professionals and the general public. And we both use national and international recognized guidelines with the Ministry of Health, Cancer Research UK, American Cancer Society as some of the organizations that we use recognized guidelines from. The areas or topics covered by the CIS are general knowledge about cancer treatment and its side effects, diet and mental health support services, which are so important. It's a database as well for hospitals, home nursing, palliative care and support groups, as well as financial assistance, looking at what is available. So we assist in terms of navigating services such as insurance, EPF, SOXO, and spiritual bodies, religious bodies, NGO funds, and governments. And lastly, it's a patient navigation program. So we can actively track people under our system who have cancer to ensure that they have the support necessary to continue their journey and connecting them to clinical trials if required. So this is the Cancer Information Service, our CIS. The helpline is 1-800-88-1000, help@cancer.org.my. It's free, it's multilingual, and a confidential service. So if anything you get out of this talk today is to get this number down and give it out to as many people as you can, because there's a huge amount of support that we can give people with cancer and their families and caregivers. Lastly, as I mentioned, the Children's Home of Hope. This was established in 1997, and it's to provide a comfortable accommodation for children diagnosed with cancer as they undergo treatment in HKL. So even though their treatments are in hospital, they get to walk back or get bused back to our center and stay in an environment outside of the hospital. So it's like a hotel, well, not so much a hotel, but a hostel, a home away from home, as we like to call it. And again, these services are given free of charge by NCSM. And in conjunction with the Children's Home of Hope, we do other support services, little things like a Hari Raya celebration, our Relay for Life, a fun family camp in PD, things to bring a little bit of happiness and laughter into lives of people with cancer. So what's new for NCSM in 2021? We've newly established an Adults Home of Hope, which is similar to our Children's Home of Hope, but for adults with cancer. It's located not far from HKL, and it really expanding the services that we have to support people with cancer. In conclusion, thank you for listening to me talk a little bit about National Cancer Society of Malaysia. I believe that the services that we provide and the people that are in the National Cancer Society of Malaysia are giving back a little to society. And I also believe that everyone can give back. Cancer does not just affect one person. It ripples through the family, the friends, the community. So all of us have a part to play in making Malaysia a better place for people with cancer. So thank you. Please get in touch with us at the National Cancer Society of Malaysia. If you ever need our services, if you know someone who needs our services, or if you just want to be part of our family. Thank you again. Goodbye.
Thank you, Dr. Sanjari, for the inspiring presentation. Participants, you are welcome to send your questions to Dr. Sanjari through Slido. For now, we'll go ahead with our forum session. Please welcome Dr. Priya, our organizing committee, to be the moderator for this forum. Our speakers will be answering some questions from our fellow participants from Slido, and I'll be sharing the questions right now.
Thank you to all the speakers who have spent their time to be with us today at this cancer webinar. So I will just moderate the forum by asking the questions from the participants. So I will start with the first question over here to Prof. Chong. Has it been shown that infection with a particular microorganism can help facilitate innate or adaptive immune stimulation against certain cancer antigens?
Thank you, Dr. Priya, and thank you to the participant for this question. So I believe this has to do with the immune maturation and microbe-specific immune programming. For example, it's been known that certain microbial signatures on the surface of the microbial cell wall can sort of train our immune system, both the innate and also the adaptive immune responses, which sort of creates an immune memory so that the next time we encounter similar antigens, then we could better go against that, you see. For example, the training of the human monocytes or by the beta-glucan from Candida albicans cell wall, which is, Candida albicans is an opportunistic pathogen, by the way, it's a fungal pathogen. This training induce not only sort of a memory or a better response against fungi, but also against bacteria. So coming back to your question, so research is still ongoing at the moment regarding the use of specific microbial or bacterial isolate or strains to help in cancer immunotherapy. For example, there are experiments or ongoing research done to genetically engineer some microbial isolates to drive the expression of a specific metabolite of interest, which could somehow modify the microbiota. For example, the Bacteroides strain, one of it has been modified to carry a gene to utilize porphyrin, which will help to stabilize its engraftment. This is done in mice, in animals. So this will increase the efficacy of probiotics. And it's been shown that probiotic strains like lactobacillus and bifidobacterium can serve like adjuvants to cancer immunotherapy. So I hope this answers your question. Back to Priya, thanks.
Thank you, Prof. Chong, for addressing the question. So I will probably move on to Dr. Louis. Should the general public be encouraged to take anti-cancer food like cumin and turmeric as a prevention even when they don't have risk factors? Dr. Louis?
Thank you, Dr. Priya. Yes, I think it's a very good question. So as we know, some of the natural products contain antioxidants and some essential minerals. So I think there's no harm if you are taking this natural product to enhance your immune system. So like Prof. Chong mentioned just now, some of these natural products can correct your microbiota, which may enhance your overall health. Right? So I think there's no harm in taking it. Right. Yeah, back to you.
Yeah, Dr. Lui, there's another question on the same natural products. So can a cancer patient on immunotherapy take these natural products together with the treatment? That's what someone asked.
Yeah, great question, Dr. Priya. So I think Dr. Ho can answer that because he's using PD-1 to treat cancer patients. But I want to caution the person who asked this question that there are some natural products that can suppress the immune system, right? So we don't know whether beta cumin, right, contains any active substance that can suppress the immune system, right? So you have to check with your local doctors what any suppressor effect from these natural products. Right.
Thank you for addressing that. So we will move on to Prof. Ho. In the study on APOBEC3 breast cancer patients who responded to Pembro, what were the PD-L1 tumor proportion scores?
So the PD-L1 tumor proportion score is a different biomarker versus the APOBEC3B mutation. People are looking for biomarkers that predict immune response. So like microsatellite instability tumor or tumor mutational burden, more than 10 mutations per megabase pair. So APOBEC3B mutation makes a tumor with hyper mutation. It makes the tumor to carry more mutations, mutational burden, therefore more immunogenic. PD-L1 score is good for certain cancers like lung cancer or esophageal cancer, but it is not so good for other cancer like melanoma or renal cancer or gastric cancer actually is good. So basically different tumors seems to have different biomarkers. In the oral study, we have not had because it's still recruiting, we do not have the clear idea of the relationship between PD-L1 score and APOBEC3B mutation, but that would be something we'll look at in the future.
Thank you, Prof. Ho, for addressing that too. We will now move on to Prof. Yu Ming Wang. There are two questions here. What are the chances of using PBT for brain cancer? Which is more effective than gamma knife? And the second question is, will PBT become a more generalized form of treatment for late-stage cancers? And are other institutions planning on using this approach as well? So to you, Prof. Yu.
Okay, thank you, Professor Priya. And and let me answer the first question. And actually, I prepared some more slides to explain this. So may I share my screen? Okay,
Yes, sure.
I will do that. Okay, so you see that on your screen?
Yes, we do.
Okay, so for brain cancer or for brain tumor, yes, we did treated patients with a brain tumor, meningiomas, glioblastoma, astrocytoma, and even pediatric brain cancers. But I was not able to share all these results because there's a lot of things to share. And I think the benefits is that the normal brain would be preserved, and or we say less damaged by proton beam therapy compared to conventional photon radiotherapy to treat the tumor or the cancer. I think this one is a meningioma. And for the left side is photon radiotherapy. So red color is the dose we need to treat a tumor. But just like the rainbow, the yellow, green, or purple means the low dose area, that's which is unnecessary dose that we don't want that to go into our brain. So you can see that for proton beam therapy, the dose is very condensed to very precise to the tumor area, and without low dose to the brainstem, to the optic nerves, chiasm, and to the rest of the normal brain. But for photon radiotherapy, the dose goes like this. So the brainstem, optic nerves, or the normal brain will be more damaged. So for patients who seek to have better preservation of normal brain, I think it is a choice. And in terms of gamma knife, okay, let me stop sharing this. Okay. So in terms of gamma knife, oh, I'm sorry. So, okay, so in terms of gamma knife, I think the effectiveness would be relied highly dependent on the dose. And yes, we can do a proton beam therapy in a single or a few shots. And that's what we did. And okay, for the second question. Okay, and for the second question, actually it's getting popular worldwide in the US, in Europe, or even in Asia. But the biggest issue for a proton beam therapy is the cost to build a proton center. For our center, our institution, actually we spent around 170 million US dollars and spent around 5 years for the construction. So I think for the proton centers, the biggest issue is the sustainability. That's all my answer. Okay, thank you.
Thank you very much, Prof. Wang. Thank you for sharing those information. It was really good findings from the study. And we will move on to the other questions. Now we have to Prof. Chong. Will taking probiotics like Yakult or Lactoforte help in improving response to immunotherapy such as Keytruda besides taking high fiber diet as probiotics? Prof. Chong, you're muted.
Okay, thanks, Priya. Thanks for the question. As I mentioned briefly just now, in general, probiotic bacteria like the lactobacillus species and the bifidobacterium species have been shown to positively impact anti-cancer response. And they can serve as adjuvants to boost immunotherapy efficacy. For example, I think there's a study showing that Lactobacillus caffeae could enhance the expression of co-stimulation and maturation markers for dendritic cells, and also activating the CD8 plus T cells. These will mediate the T cell mediated killing of cancer cells. And also not only that, the metabolites produced by some of these probiotic bacteria like vitamin B6 was shown to have synergistic effect when combined with cisplatin for treating the non-small cell lung cancer. So yeah, I think overall taking probiotics do help. Okay, back to you. Thanks, Priya.
Thank you, Prof. Chong, for answering that question. The next question is to Prof. Ho. What explains the synergistic effect from combining chemo and immunotherapy?
The relationship between chemo and immunotherapy is not so clear. So we hope that it is synergistic effect, but it may also be additive effect. But there are some theoretical basis why adding chemotherapy to immunotherapy will help. One would be you kill off more of the tumor cells therefore generate more tumor-associated antigens that the antigen-presenting cells can pick up. The other benefit is that the immune system works with check and balances. So in a lot of tumors, there are lots of regulatory T cells, so and myeloid-derived suppressor cells. So giving some chemo before immunotherapy may help to kill off some of the regulatory T cells and myeloid-derived myeloid-derived suppressor cells, so that the T cells can work better without inhibition. But there are other ways to achieve synergistic effect, like radiation or some agonistic antibody. People are looking for the combination treatment, and this is still an ongoing search for that.
Thank you, Prof. Ho, for answering that question. So we move on. The next two questions are for Dato' Saun. What is your opinion regarding the use of blood tumor markers for cancer screening in healthy people that is widely offered by clinics or hospitals here?
Thank you for that question. We actually don't encourage the use of blood tumor markers for cancer screening. It is not a globally accepted tool for cancer screening. It is something which is used more for detection or in terms of treatment. So to be honest, we're very much against the use of blood tumor markers, especially because they're non-specific. And they're also done in a manner where the patient is not actually counseled beforehand on the impact of elevated tumor marker. There are many people who've had elevated tumor markers, which then make them believe that they have a cancer, and will continue to do investigations for cancer when actually they are healthy individuals. So I do caution in terms of doing blood tumor markers. The only tumor marker which is indicated for cancer screening is the PSA, prostate-specific antigen. And even that, in most countries now, it is not done widely without prior counseling of the patient on what a PSA might mean to them.
Yeah, all right, thank you, Dato'. And the next question also to Dato' Saun is, how can the staff and students from university work with the National Cancer Society of Malaysia?
I love that question because it means that people want to work with us. You can work with us in different ways. A lot of student organizations and universities come to us to do community projects within the university itself. So actually raising awareness on prevention and early detection of cancer. So that's one way, is, elevating the cancer awareness within your own university and student bodies. The other ways, though, are more specific. We have universities work with us on internships. They also come and work with us on research projects. Much of this is linked to community activities that we do, implementation research that is done. And it's a lot of it is on behavior, on the behavioral aspects of people with cancer, or on the general public in terms of their perception of cancer. And of course, the last way of coming to us is, if as an individual you want to volunteer with the National Cancer Society of Malaysia, we're very, very happy to.
Thank you, Dr. Saun. So we will be in touch with you for that. Okay, so we will move on to Dr. Louis. The next question is it possible that some supplements or drugs that promote Treg activity can potentially reduce the efficacy of immunotherapy treatment?
Thank you, Priya. It's a very good question. The answer is yes. So as Dr. Ho explained previously, before the patient start immunotherapy, sometimes they are given chemotherapy. So the reason is because they want to remove the Tregs and the myeloid-derived suppressive cells before starting the immunotherapy. Because Treg is a brake in the immune system. So once you remove these brakes, the immune system can be activated, but there is a downside where the immune system will be overly activated, and this will cause the autoimmune disease. Back to you, Dr. Priya.
Okay, thank you, Dr. Lui. There's another question also to you. Will the bitter cumin compound produce the same effect on normal cells?
Thanks, Priya. Again, a good question. So the vernodidine cannot differentiate between a normal cells and a cancer cells. So when this compound goes into the cell, it will activate FOXO3A and cause apoptosis, regardless whether they are cancer cells or normal cells. So the good thing is we are betting that the cancer cells will be more susceptible to the killing effect of vernodidine because they are quite intoxicated to the oncogene compared to the normal cells. So yeah, it will produce the same effects, but at a different degree, I would say. Thank you.
Thank you very much for addressing that. Next question is to Prof. Chong. How much will the effect of opportunistic bacteria in the gut have on immunotherapy for cancer, and how to manage around it?
Okay, this is a good question. So far, large randomized clinical trials.
Prof. Chong, would you be able to speak a little louder? Your voice is sinking.
Okay, sorry. Is it better now?
Yes, yes.
Okay. So far, large-scale randomized clinical trials on the cancer immunotherapy have not revealed any higher risk of infections. But in the cases where patients have immune-related adverse events, then they will have to undergo immunosuppressant therapy. For example, corticosteroids have to be instituted. So if you have corticosteroids, this could lead to opportunistic infections. Having said that, there are also sporadic cases or some cases of patients undergoing the immune checkpoint therapy who have their latent or dormant infections by bacteria being reactivated. For example, like TB, tuberculosis, and also some viruses like hepatitis B and C and the varicella zoster virus. So in such cases, if these opportunistic latent infections are activated, then of course, I think the way to manage is to administer the exact specific treatment, like anti-TB treatment for tuberculosis. So I hope this answers the question. Back to you, Priya.
Thank you, Prof. Chong, for addressing this question. Next question we have is to Professor Wang. How does the cost of PBT compare to something like Gamma knife?
Okay, thanks, Priya. And actually, I have to clarify the cost. What does the cost mean? It means to build a proton center versus to build a gamma knife center, or the cost for treatment. And I believe maybe she or he is asking about the cost for treatment. Okay, and unfortunately, proton beam therapy here in Taiwan is not reimbursed by our national health insurance. Taiwan has a public health insurance system we call National Health Insurance. So for proton beam therapy, we need to patients need to pay out of pocket money. But for gamma knife, some patients they can apply for national health insurance, so they won't need to pay. So I think that makes a big difference.
Okay, all right. Thank you for addressing that. And there are last two questions. One is to Prof. Ho. For MET polysomy in lung cancer, would you expect response to MET targeted treatment, even though it targets mainly MET amplification or deletions? Prof. Ho?
There are many types of MET mutation and overexpression. So the main one is actually the exon 14 skipping mutation, but they found out the secondary mutation after the patient has have taken EGFR inhibitor, then you can get MET overexpression. So people have looked at the response to different type of MET mutation. I think the most likely to respond would be the exon 14 skipping mutation and the MET amplification, which is a secondary mutation. And now there is actually MET inhibitor study in Malaysia. My colleague, Professor Liam, is running the INSIGHT 2 study for patients who have failed first-line osimertinib. So they rebiopsy the patient, look for MET amplification, and put patient on tepotinib if they have the mutation. So MET inhibitor is coming up.
Thank you, Prof. Ho, for the information. Next question is to Datuk Soundri. How can the urban poor be made aware of the work of National Cancer Society?
Thanks. It is difficult, I have to admit, to get our name out there for many people. But the way we work is that we try to put ourselves out in national media and social media, etc. But the other way of doing it is actually working with community leaders, and that's what we do. So we work with various community leaders, be it on the district level or be it on even on the religious level, to work with them in terms of doing community projects with them or having clinics out at the urban poor areas. And also to pull them in for our Etika free mammogram campaign. So, you know, for those who want to work with the urban poor or who do know organizations who work with the urban poor, we'd really like to link up with them to see what we can do with them as well.
Thank you, Dr. Sound. That sounds great. So I'm sure there will be many others who will be also in touch with you with such work to be done. So I think we have ended the forum. There are no more questions for the speakers. I would like to pass it back to Dolisha, our MC. She has some announcements to be made as well.
Okay, thank you, Dr. Priya. Okay. So thank you all speakers and Dr. Priya as our moderator for the forum session. So for the moving right along, we want to thank you, our organizing committee, for sparing your time arranging and organizing this event. Special thanks to Prof. Rusli, Dr. Wong Yin Hao, Dr. Sapna, Dr. Yong Chai Hong, Dr. Priya, and Dr. Lim Su In, and all of our Taylor's Medical Society committee for the well-organized webinar. I apologize on behalf of Prof. Rusli as he can't join us today due to an unpredictable emergency. He sends his regard to all and hope that our participants have benefited from this webinar. He appreciates all the speakers for the amazing presentations and is looking forward for the next collaboration. So now, for all staff of Taylor's University, a gentle reminder to complete the HRSS registration to claim 3 training hours. You may register on HRSS at the end of the webinar. So next, for those collecting CPD points, you can scan this code with your MMA app to self-register your attendance within 30 seconds. Again, for those who have not scanned, don't worry, as you will be receiving the points within the next 7 days if you have registered. In the meantime, we would like to hear about your opinion on this webinar. We would really appreciate if you can join the feedback survey in the next slide. Okay, next. So while you are still holding up your phone, you can scan this code for the post-webinar feedback survey or by clicking the form link that is set in the Google chat. If you wish to get an e-certificate for this webinar, simply tick the option at the very end of the Google form. Next. Do you know that our School of Medicine research classes are working on cancer research especially on public health research group and non-communicable diseases. Please contact Prof. Rusli for further details. Next, we will also like to announce a pleasant surprise. The Taylor's University School of Medicine Society is planning for a fundraising activity on cancer awareness. Therefore, all the profits collected from the sale of our merchandise will be channeled to the National Cancer Society of Malaysia. Do follow our Facebook and Instagram page for further information. To Dr. Santari, our team will be in touch with you soon on this. So before we end this webinar, we would like to have a photo op session with all of you. Can everyone kindly turn on your camera? We want to create a special moment with you and see those faces behind the camera. So while we are waiting, once again, we thank you for your patience and willingness to free up your time and join us on this wonderful webinar. Big thank you to our 5 great speakers, Prof. Chong Pei Pei, Dr. Lui, Dr. Ho, Dr. Yu, and Dr. Santari. Well, that's nice to see the faces that joined us for the past 3 hours. Okay, we'll have the screenshot right now. Wow, there's so much of you. Okay, just give me a minute because there's many pages to be screenshot. Okay, yeah, it's all done. Thank you all for joining us on this webinar. So, well, that's a wrap. Thank you, everyone. Wish you all a pleasant day and stay tuned for our next mental health webinar. We will update on our Facebook and Instagram. Goodbye, everyone.
Thank you.